give me one sec. Okay, I'm just going to go one step ahead, just because I'm going to explain this first and then go back. So what is happening now? Let's have a, actually, no, sorry. I'm going to talk about antigens and antibody first because I've mentioned it, but I haven't quite explained it properly to you yet, and we haven't seen sort of what's happening schematically, so let's do that first. So now we know that there are two important parts of the immune response. There is the cell-mediated immunity and there is the humoral immunity. So what happens is, firstly, we've got the antigen-antibody interactions. Like I said, the antigens, as you can see, are these receptors on the cell surface of the pathogen. So that is an antigen, and it has those, let me change the color of my pen so like I can actually use it. Oops. Okay, let's do green. So as you can see, it has this this triangle, you know, these shapes here, right? So it's got those shapes. That's the antigen. Now those shapes there, those and then those are the antigens that are found on the surface of the cell. Then we've got the antibodies, and there are three types of antibodies that you can see here, each for a different type of antigen space. They all have different antigen binding sites. So this is the antigen binding site, as you can see, and they're all different for the different types of antigen. And this process is called the clonal selection. So the process by which the presence of a certain antigen selects immune cells that are specific to it. So the antigen, so the antibody that is selected for the antigen is going to be specific to the antigen. And this process gives us the adaptive immunity response that is specific to the pathogen. And this is like evolution, but for B and T cells. So what happens is that we've got the antigen molecules. These are the antigen molecules, and the B cells that differ in antigen specificity. They all have different shapes, as you can see here. This one has triangle. This one has like triangle poking upwards. This is like a V shape here, and this one is like a U shape. So they're all different antigen receptors. And what happens is, sorry, these are all different antibodies. Sorry, and what happens is when they when it sees the antigen, it tries to match up. It tries to match up and find the perfect match for whatever the antigen molecule is. And whichever one fits, so in this case, this specific one fit to the antigen. And so then this cell will replicate. The plasma B cells will make lots of copies of the antibodies, and those antibodies will go ahead and they will stick to that pathogen. And what also happens is that we get lots of clones of the memory cell, so that in our body later these memory cells can circulate in case we get a reinfection. They can quickly go ahead and deal with it. And the clone of the plasma cells, these plasma cells produce the antibodies, which then come off the plasma cells, and they go ahead and latch onto the antigens wherever they find the antigens in the body. So what happens now is, I'm going to go one step ahead. What happens, actually no, this is actually, it would make more sense to go this way. Sorry, I'm like skipping ahead in my own mind, so we'll go through this first. So this is the process now, where I introduce you to a concept called the major histocompatibility complex. So every single cell, so basically present on every single cell surface, it's specifically reserved for the presentation of molecules such as antigens and identification. So there's a histocompatibility complex that is found on the cell surface of every cell, and it is used to either signal an antigen or for identification, so so that your body is like able to distinguish between self and non-self cells. So there is two types of histocompatibility complex. The first one is present on all nucleated cells, and it presents the normal proteins it makes and the abnormal ones. So what that means is these are the normal cells. It shows the immune cells that, okay, these are the normal cells. MHC2 is only present on antigen presenting cells, and those are known as the APC. So they're only present on the antigen presenting cells. And what happens is they present the abnormal pathogenic antigens. So you can see over here that we've got in all nucleated cells, this is MH class 1, we've got the antigen binding cleft, and this is the beta-2 microglobulin, so it is part of the body, so it latches onto here. This is for all nucleated cells. But in MHC class 2, there's an antigen binding cleft, right? But here we've got the antigen itself, so beta-1, beta-2, alpha-1, and alpha-2. So what does the major histocompatibility complex do? Now I'm going to take you for a second into the world of my favorite books. If you're not very familiar with it, I will try my very best to explain it, but hopefully most of us would know what's going on. So we've got Death Eaters. The Death Eaters are looking out in the Prisoner of Azkaban in book 3, they are looking out for Voldemort. Right, so they are looking out for Voldemort, and as a result of that, they're going to stay away from the students, but they're looking out for anyone who resembles Voldemort. And as a result of that, everyone else has to be careful not to be like near Death Eaters, because once they get to a person, like they can suck the person's soul out. That is the whole point of a Death Eater, Death Eater's kiss. And also, I want you to imagine now that the Death Eater in this case is the cytotoxic killer T cell and the natural killer cells. Like I explained to you earlier, the cytotoxic killer T cells is that destructive cell in the group, that angry young cell, which is very hot-headed. And this cell, what it's going to do is it is going to be on the lookout for antigens. If it sees an antigen on a cell surface, it is going to go ahead and kill that cell off so that it can kill the antigen. So it's like a Death Eater in the body that is going everywhere and, like, on the lookout for a Death Eater. Sorry, and on the lookout for an antigen. And it's very dangerous. Like, if it comes near a cell, just like Death Eaters, right? If it comes near a cell, like, it can kill any cell essentially, but it is specifically looking for those cells with the antigens on it that are foreign to the body. Any healthy cell does not want to be killed by a Death Eater, is afraid of the Death Eater, is afraid of the cytotoxic killer cells, as Harry is, right? So let's say that healthy cells are represented through Harry, and that expecto patronum, that Patronus is is the MHC is the cell showing the normal proteins that are being produced on the MHC1 to actually show that, hey, I am not the antigen presenting cell. I'm not the one, I'm not the pathogen. Please let me go. That is what is happening here. So let me go through it again. The Death Eater is the cytotoxic killer cell and general natural killer cells. They are on the lookout for antigens. Any normal cell, like, they can go to any normal cell and they can kill it, but they won't because they're on the lookout of an antigen. That is the whole process of it. So that is why that healthy cell is going to show the normal proteins that it makes on its MHC class 1 to show the Death Eaters, to show the cytotoxic killer cells that, hey, I am a normal cell, let me go. That is what is happening. Harry does it. Harry produces a Patronus to drive the Death Eaters away, and in this case, the Patronus is the normal proteins that are being used. Once the cytotoxic killer T cells notice that, hey, it's the normal proteins, this is not the antigen-containing cell, they go away. So that's what the role of the major histocompatibility complex is. This is the analogy to help you understand it. So let us now focus on the big idea of what is actually happening. So I've talked about what happens in the cells. I have talked about basically, I've talked about what is happening. I have talked about the big idea of the individual cells. Now let us look at what happens as a whole. So let me walk you through this process. I'm going to make sure that my annotations are not messy, that you can read this. So let me walk you through this process and follow along with me. These slides are here for you. If you want to go ahead later and like write down step by step what happens, that would be really helpful, I believe. So we've got a pathogen, right? That's our pathogen. And this is the antigen that is being presented on its surface. Every cell will present an antigen on the surface. Normal cells present like the proteins they make to show that they are cells, right? Like our normal cells will show our immune systems that, hey, we belong to this body by showing us the proteins on the MHC class 1. With pathogens, they are going to show the antigens. So once the body sees this, what happens is these pathogens are immediately engulfed by macrophages. And macrophages, I cannot read that, but I believe that is, they have the MHC. So they're able to see what the antigen is here, basically. So these macrophages go ahead and they engulf the pathogen like the macrophages do. Then once they engulf the pathogen, they present the antigens on their surface. So this is where now, this is the MHC class 2 presenters, basically. These are the MHC2 class 2 presenters. So what happens is they eat the pathogen up and then they present the antigens on their surface. Right there. And then interleukins, which are like proteins in the body, they call helper T cells and B cells to the site of infections. Then macrophages, they present these antigens here, and basically we've got antibody mediated immunity here and cell body mediated immunity here. And what happens is, let me change my ink color. And what happens is with the helper T cells, so they've got the T cell receptors here, and they latch on to the antigen here, and same thing happens with the B cells. The B cell will then differentiate into plasma cells and memory B cells. The memory B cells are going to store, they're going to be stored in the lymph nodes, and the next time that this pathogen comes back, they're going to be circulating and immediately jump to action. With plasma B cells, this antibody fits the macrophage over here. Remember, this is why it takes time in that first process for the body to find the right antibody for the antigen. The process is slow because we're trying to find out which antibody, because we have lots of antibodies, we are trying to find out which one fits the antigen that is on the surface. So essentially, once it differentiates, the plasma cell clones and they let go and they secrete all of these antibodies. These antibodies then go ahead and immobilize these pathogens. They go to the pathogen and latch onto the pathogen. And what this whole process does is, now the fact that the antibodies have latched onto the antigen, the body knows that it's flagged to the body that these guys are foreign invaders. We don't want them. That is why this whole process happens, because we need to know what that antigen is that we're trying to kill. And so these antibodies latch on to those antigens. So, and once that happens, our T cells come into play. So helper T cell has gone ahead, got the antibody, right? And then it has taken the antibody, it is stored in the memory B cells. These are the smart guys like the memory B cells here, and they remember that the next time that we see this antibody, this antigen again, this is the antibody that works. We also have, we also see the helper T cells send out cytokines, like I said, and those cytokines call the killer T cells and suppressor T cells. The killer T cells comes and kills the pathogen that has the antibody attached to it, right? And it secretes perforin, which is a toxic substance to destroy the pathogen. And what happens is our suppressor T cell comes along as well, because it is the killer T cell's best friend, and it comes along and it suppresses the killer T cells, tells it to calm down once we have finished the immune response, once we have destroyed the pathogen. Because remember, it can't keep the immune response going, otherwise that becomes destructive. The killer T cells can essentially kill all the cells, and they can affect all the cells. And we have obviously seen autoimmune diseases where the body doesn't recognize the self cells as self, and they go ahead and destroy those cells and don't let them function. The regulation is all messed up. So that can happen. That is why we see these cells working together, and the suppressor T cells and the suppressor T cell suppressing and regulating the activity of the killer T cell. So this whole process starts off with the pathogen, with the antigen. The macrophages come. These are our special white blood cells. They come, they display the antigen on the MHC, wait, let me write that down, MHC2 receptors, because those are antigen presenting. Right there, these yellow thingies, right? And then after that, they go ahead and they call the helper T cells and B cells. And once these cells come, they make these antibodies that can then go ahead and latch on to all the antigens, all the pathogens. You know, the pathogen has replicated by now, so go ahead, latch on to the pathogen and deal with it. That is the whole process. So I hope this was understandable, and definitely I would suggest going ahead and watching some YouTube videos, because they really help me understand the whole process. Amoeba Sisters is a good channel. They make YouTube videos which is like generally at our HSC level. Also Crash Course, of course, and they make awesome videos as well. Like the visualization is really good in both of those videos, and if there are any other good ones that you can find out there that don't go into too much detail, but like this is the kind of detail